So once again, good afternoon everyone. To our teacher, good afternoon ma'am. So now I am going to present or I am going to report the side chain bases for the nucleic acid, which is the continuation of the report of Mr. Joyre Bituonon. So when we say side chains bases, or usually called bases because they are weakly basic. So there are five major types compared to the 20 amino acids or the three pyrimidines, which is the cytosine, the thymine, the uracil, and among one six-membered ring. So T appears in DNA and U appears in the RNA. So there are two purines, the adenine and guanine or guanine. So the two rings, six-membered ring fused to a five-member ring. So in thymine, first, sugar atom numbers are denoted with a prime to distinguish them from purine or pyrimidine atom numbers. So site of attachment to furanous, the C1 or the glycosidic bond. So in adenine, the purines, guanine, the cytosine, thymine or DNA pyrimidine, and uracil for RNA. So in DNA base pairing, in Watson-Crick base pairing, adenine or adenine or a hydrogen bond with thymine and guanine. So hydrogen bond with cytosine as shown. So as you can see here in the hydrogen bonding for planar sp2, the hydrogen bonding which is the guanine and the cytosine. So in here, adenine and thymine. So in contrast, non-Watson-Crick or wobble base pairs involve complex structure. Example, A plus C. So non-canonical pairing is rare but can lead to point mutations. So in DNA is a double helix. When we say a duplex is formed between two strands of DNA with complementary bases running in opposite direction, which is the TG or the thymine and guanine, adenine and cytosine. For another one is ACCTG. So double-stranded DNA forms a right-handed twist like the threads on a bolt. 3.4A between vertically stacked bases, helical repeat distance of 34 to 36A. So 10 base pairs in each turn. It contains a major groove. Base pair functional groups are exposed sequence-specific DNA binding proteins, transcription factors, recognize the major groups zinc, finger, helix, turn helix, or leucine zipper domain. And a minor groove base pairs are more protected by the phosphate sugar backbone. Bind structural proteins or non-specific bonding that package DNA into compact chromosome. So in this part, the higher level is the minor group from the double helix, and in the middle part is the major group. So now, model of DNA first proposed from three data, which is the Chargaff's or rules. The A is equal to T, G is equal to C, or the pyrimidine is equal to purine. So base are relatively inaccessible and not titratable. According to Rosalind's X-ray diffraction image, which is proponent by Rosalind Franklin 1920 to 1958. In 1962, Nobel Prize Medicine to Watson Crick Wilkins. So X shape in here indicates the helix spacing, which is have a 3.4 and 34A periodicity in long axis. So, double helix papers. So, mm, the father of molecular biology, the Linus Pauling 1901 to 1994, sickle cell disease due to mutation in specific protein, according to him. And he proposed the alpha helix and beta sheet as the main elements of secondary structure in protein. So in DNA is a double helix or secondary structure. The Watson-Crick structure is called B form DNA. So most stable structure for random sequence DNA under physiological conditions, regular repeating structure can support long lengths, compact packaging by coiling DNA around spindle proteins, histones, good information storage medium. So other types of DNA or the double helices, A form occurs when water is lacking, wider diameter than B form. So both right-handed. So another one is the Z form, which is particular to certain sequence, or the left-handed narrow diameter. So RNA transferring information. So the transferring information from RNA takes in many structure, from DNA to RNA transcription. Messenger is the RNA, the to mRNA is synthesized on one single strand DNA template in the nucleus by RNA polymerase. When we say RNA, which is the protein and translation, mRNA exits the nucleus and associates with ribosomes in the cytoplasm. The mRNA plus ribosome recruits transfer RNA to tRNA, covalently bound to an amino acid at its hydroxyl. So three adjacent bases on mRNA, which is the codon, implement three adjacent bases onto tRNA and on tRNA, which is the anti-codon. Every 3 liter of the DNA sequence correspond to an amino acid. So mRNA streams through the ribosomes with its new tRNA that is recruited. A new amino acid is added to growing polypeptide chain through the formation of peptide bond. So this is the formation of the nucleosome or quaternary structure. The short region of DNA double helix, which is 2 meter, beads on string form of chromatin, which is 11 nm, 30 nm for chromatin fiber of packed nucleosomes, 30 nm, section of chromosomes in an extend form, 30 nm, condensed section of chromosome, 700 nm. So entire mitotic chromosomes and centromere is 1400 nm. So the nucleosomes, histogenes are basic proteins that bind strongly to DNA. They help to organize and compact DNA in the cell nucleus. They form nucleosomes, regular units of about 200 base pairs of DNA wound around an octameric core of different histones proteins. Based on a string, nucleosome can be packed together to form a coiled fiber, and it conserved license can be acetylated or post-translational modification. So to loosen chromatin packing, amide is not charged and no longer attracted to phosphate and promote gene transcription. So the pattern of chromatin packing can be transferred to offspring or epigenetics. So this is the basis of innate memory and tissue differentiation during development. So other carcinogens in food, the browning of bread, meat, and roasted coffee get their odor and flavor from a mixture of compounds that result from amino acids reacting with sugar at elevated temperature. So the Maillard and this is called Maillard reaction. So the sequence of events starts with the nucleophilic amino group or enhanced in basic solution attacking the sugar carbonyl group to form a glycosylamine or BN glycoside. So eventual product include cancer-causing acrylamide and furans. So the Maillard reaction also takes place spontaneously in human tissue and its products are linked to diabetes and cataracts. So the key concept map for DNA structure which is from DNA. So the DNA structure consists of the deoxyribose, the phosphate in diester linkage, so bases, the adenine, thymine, cytosine, and guanine. So the phosphate in diester linkage form the deoxyribonucleotides. The deoxyribonucleotides form a double-stranded helical molecule and in which form antiparallel that create polarity. So in which the nucleotide sequence can be 5' end to the 3' end. So another form or it becomes stabilized by the double stranded helical molecule can be stabilized by hydrophobic interaction between stack bases and the hydrogen bond between base pair. So also it can become a single stranded. So for hydrogen bonds between base pair, it's specifically which is the adenine which pairs with the thymine and cytosine pair by guanine. So the single stranded as a result of the replication and denaturation. So another key concept map for an RNA structure. So RNA consists of ribose, phosphate in diester linkage, the bases which is consists of adenine, uracil, cytosine, and guanine. So the phosphate in diester linkage produce the ribonucleotides and the ribonucleotides polymers of which form the tRNA. So another one, the ribonucleotides can form the RNA and the eukaryotic mRNA. So the mRNA can structural features include the 3 A tail, the 5 A cap of 7-methyl guanosines, and the monocistronic. So the functions as template for protein synthesis. So the tRNA can be structural features include unusual bases, extensive intrachain base pairing, at least one specific type of molecule for each of the 20 amino acids found in proteins, which is the 3-CCA. So another one, the function of tRNA is the adaptor molecule that carries a specific amino acid to the ribosome or the mRNA complex. So, the structure for rRNA include associates with protein, the 3 size species in prokaryotes, 4 size species in eukaryotes, and the last one is the modified ribose and bases. So the function of these are structural component of ribosome. So I hope you learned something today, and that's the end of my report. Thank you for listening.